Type 1 hypersensitivity reaction. We do having type 1, type 2, type 3, type 4 hypersensitivity reaction. So the type 1 hypersensitivity reaction is also called atopic or anaphylactic. Now this is type 1 hypersensitivity reaction is also called immediate hypersensitivity reaction, because the response of type 1 hypersensitivity reactions occur within minutes. That's why type 1 hypersensitivity reaction is also called immediate hypersensitivity reaction. Now the antibody for type 1 hypersensitivity reaction is IgE. Now I already explained in eosinophil what are the function of IgE. Okay, so IgE, first of all, it's bind with basophil and mast cells. There are Fc receptor. There are receptor on this mast cell and basophils with which this IgE bound. Okay, are bind. Now the second function of IgE is lead to a coating of parasite, and as it coat the parasite, then it kill the parasite. Okay. Now these are the two functions of IgE. So the antibody of type 1 hypersensitivity reaction is IgE. Now the cells, there are two effector cells of type 1 hypersensitivity reaction. The one is called basophil, which in the circulation, and the another one is called mast cells, which is the location for it is in tissue, epithelium and mucosa. Please watch my last videos on basophil and mast cell. Now the complement system, there is no complement system involvement. There will be different phases of type 1 hypersensitivity reactions. Okay, so the phases. Now there are two phases. The one is called phase 1, which is a primary response, and the another one is called phase 2, which is a secondary response. In the primary response, there will be first contact of the allergen. Okay, if this is a person, if he for the first time he just exposed to the allergen will come into a body, and this allergen will be phagocytosis by the macrophage, and macrophage is antigen presenting cell. I will explain in next video what are the antigen presenting cells. So it will take this allergen, and this will form a phagosome, and then lysosome will come, and that will be called phagolysosomes. Okay, then this phagolysosomes will digest this allergen, and this allergen will convert into small particle epitope. Okay, you can say epitope or whatever. Now this and the MHC2 molecules will bind with this epitopes or small particle of the allergen, and it will be expressed on the surface of macrophage or antigen presenting cells. Now this is MHC2 molecule. As it come in contact with the T helper cell, it will activate the T helper cell, and as we activate the T helper cells, now the T helper cells is going to convert into T. This is naive T cells, and this naive T cell will be activated, and this naive T cells have the ability to convert into T helper 2, and also the ability to convert into T helper 1. But here this naive T cells is going to convert into T helper 2. So the allergen is going to activate the T helper 2, and this T helper 2 T cells is going to secrete interleukin 4. And this interleukin 4 is going to, because it will activate the class switching, like the B cells is going to convert into plasma cell, and then plasma cells are going to produce antibodies. Okay, but interleukin 4 is for IgE antibody. So this is for MCQs, you have to remember interleukin 4 is for the production of antibodies IgE. So there will be the production of IgE. Now this IgE have a very short half-life. So this IgE will come, and the basophil and mast cells, they have the basophil and mast cell surface, we having this Fc receptor. You see this is Fc receptor, and okay, and this is the Fc portion of IgE. So the Fc portion of IgE is going to bind with this Fc receptor. So now you see this phase is sensitizing phase. Now this is for the first time, if the exposure is occurred, this is called sensitizing phase. Okay, so now the person is going to be sensitized, and now this was the primary phase. In the primary phase is asymptomatic. You have to remember the primary phase is asymptomatic. Okay, and the primary phase is also called sensitizing phase. Now this IgE bound with the mast cells and basophil. Now if in the secondary phase, if the allergen re-enter into body, into the body, you see IgE was bound on the mast cell surface in the primary phase. Now in the secondary phase, if this IgE re-enter and after re-entry, now this allergen will directly bound with, okay, will directly bound with IgE, and as it bound with one IgE and another IgE, there will be cross-linking will be occur. You see, this is allergen, this is allergen, this is allergen. Now the cross-linking of allergen is the cross-linking of IgE occur. Now this allergen is going to connect the one IgE to another IgE, and because of this, the mast cell degranulation will occur. And after the mast cells degranulation, there will be secretion of histamine. You see the histamine secretion is occur now. Now this histamine, as I already explained in the last video, please watch my video on mast cells, okay? And there I explained that then this histamine is cause vasodilation, okay? This histamine cause vasodilation, and because of vasodilation, there will be increase in blood flow, and because of blood flow, okay, now this will come in the sign and symptom of type 1 hypersensitivity reaction. Histamine will cause vasodilation. Because of vasodilation, we increase the blood flow, and if the blood flow is increased, that area there will be redness, there will be flushing, okay, and there will be warm. And as you increase the vasodilation, there will be increased vascular permeability, so the vascular leakage will occur. The fluid will move from the blood vessel into nearby area, and that will lead to swelling or urticaria, which is swell area on the skin. And if we also increase the blood flow, there will be decrease in blood velocity. So if you see here, this one blood vessels and this one blood vessels, here the diameter is less, and here the diameter is increased. So if we increase the diameter, blood flow will increase, but the velocity, the movement of blood will decrease as the diameter is increased. And because of that, blood stasis can be occur, which can form the blood clot, okay? Now what are the another function of histamine, okay? So you see here, this histamine, first of all, you already know that histamine will lead to vasodilation. Now the second thing you have to understand about the histamine, that there are H1 receptor and there are H2 receptor in our body for histamine. So you see the H1 receptor, it will lead to smooth muscle contraction, and the smooth muscle are present on our gastrointestinal system and respiratory system. In the gastrointestinal system, if the smooth muscle contraction occur, that will lead to constipation and cramp. If the smooth muscle contraction in our respiratory bronchiole occur, that will lead to asthma, a shortness of breath, and it will also increase the production of mucus in nasal mucosa, and that will lead to congestion. And if this is a sensory nerve in here, we will having histamine, and that histamine can lead to itching and pain in our body, okay? So hope you will understand this. Now in the secondary phase, now we have to understand what are the example of type 1 hypersensitivity reaction. So the example, as I told you guys, if we see this A in the first, that will be type 1 hypersensitivity reaction, like atopic disease, asthma, allergic rhinitis, atopic dermatitis, allergy. We are having allergy to food, animal product, anaphylaxis to drugs. Sometimes the drugs can also cause the type 1 hypersensitivity reaction. And high fever, and insects like bee, the bee venom containing melatonin, which also lead to mast cell degranulation. After mast cell degranulation, there will be secretion of histamine. Then this histamine will lead to the all sign and symptom of the type 1 hypersensitivity reaction. Please watch my video on mast cells, so there you can understand the full concept of histamine. And you also have to understand that what are the causes of degranulations of the mast cells. So IgE, which we already explained here, and anaphylatoxin C3a, C5a, and interleukin 8 can also lead to cause degranulation of mast cells,